Hi everyone, today's pre-lab video is going to be about blood typing. So welcome to the office today instead of the front door. So let's talk about blood typing. So blood typing has an interesting characteristic in that it has multiple alleles. It doesn't just have a plain dominant or recessive. There are actually 3 alleles, and we'll talk about those in a second. But blood typing is kind of special because it shows a characteristic called codominance. And by the name, you can probably guess what codominance means. Co means together, and dominance means dominance. So together, dominant. It means that 2 alleles can express at the same time. So instead of one masking the other, we actually see both of them at the same time. And it can be fairly common in humans, but this is our simplest example of that in blood typing. So the exact definition of codominance is that the heterozygote so 2 different alleles expresses more than one allele at the same time. So we don't have this masking effect where one is more dominant than the other. We actually see both equally. So if you wanted to see that, this definition is on page 279 in section 9.8. It's 9.9, sorry, section 9.9. Page 279, that's the definition of codominance. So if you move forward and talk about blood type, let's talk about that now. So you probably already know that there are 4 different blood types. We have A, B, AB, and O blood types. And we can also call these blood groups. And the individuals who carry these have different characteristics within their immune system and with the red blood cells themselves. Here's where our red blood cell drawings of a long time ago actually might come in handy. So we did lots of those a while back. And so the fact that we have AB, and O blood types, it affects who can give blood and who can receive blood of different types. So the characteristic that we have here for blood groups, we have something called multiple allelism. Multiple allelism is actually meaning that there is not just dominant or recessive, like I said in the previous slide. We have more than 2. In this case, we have 3 alleles. So that's what that means. Multiple allelism means a gene has more than 2 alleles. Now this is not unusual at all. This is very common. So the difference though is that we still only have 2 alleles within our cells. You could say we only carry 2 alleles on the homologous chromosomes. Right? Because we have mom chromosome and dad chromosome, and we can either carry, we have one here and one here, but in a population we will see more than just 2. So we actually have 3 different blood alleles, and those alleles are, we have what we call IA, IB, and I. So IA is a capital letter, so it's a dominant trait. We have IB, which is also a dominant trait, and we have little I. I'm going to do a kind of fancy little I, just because that's what I'm used to. So IA and IB are dominant, and little I is recessive. So we can see that the word codominance would work with this, because if we have IA and IB together, we see the AB blood type. We'll do those genotypes in just a second. So IA, IB are dominant, and little I is recessive to both of those. So let's talk about the genotypes. If we have type A blood, so let's draw some little diagrams here. If we have type A blood, I'm going to do a grid here. I'm going to do it in fourths, the best I can here. Ooh, a little crooked, that's okay. So let's start up at the top, and we have type A, type B, type AB, and type O. All right, so let's draw some blood cells. I'm just going to go ahead and draw all of them. All right, there's my blood cells. So type A blood has what we call A antigens on the surface. Now these antigens are recognition proteins. If you remember back from the membrane, they are part of the cell identification. Now type B, we can imagine, has something we call type B antigens. Now notice they kind of look like A and B here, with the humps versus the spikes. It's to help jog your memory. Type B has B antigens. And type AB has, guess what, A and B antigens on the surface of the red blood cells. And type O has no antigens on its surface. So let's think about the genotypes here. We've got our phenotypes, which are the production of the antigens, but the genotypes are slightly different. So to have type A blood, you can have, remember that we have two alleles because we have one mom chromosome and one dad chromosome. We can have IA, or IA and little I. Little I because it's recessive. Remember that the IA will mask it completely. In the B type blood, we can have IB, or IB little I. Just like that. Just like A. Now type AB blood has to have IA, IB. Doesn't matter what order they're in, but I do AB. And type O has little I, because the little I does not code for any sort of antigens on the surface. All right, let's see. So let's talk about antibodies for a second. Let's go on to the next page here. So we have these blood types, right? So we have antibodies also that are produced by our cells in order to prevent foreign blood from being given to us. So if we make kind of a, ooh, that's bad, kind of a grid like before, can I get it straighter? I can, sort of. It'll do. kind of hard to angle the tablet. I'm just going to put A, B, and O. So let's talk about antibodies. Antibodies are produced by the immune system, and they will actually attack foreign things. They're what causes you to react when you have a cold or the flu, and causes your immune system to act. So we have different types of antibodies. Remember that A produces A antigens. Well, it produces also something called B antibodies. I'm going to do them kind of like this, because the B antigens I make round. So they make B antibodies that actually attack the B antigens. That's why we can't give B blood to A patients or vice versa. Right? Don't want that. And it actually causes the blood to clump up or agglutinate, and it can cause clots or even death over time. It's really not good. Now, type B blood individuals make A antigens, or A antibodies against A that will attack if they're put in the same area together, and they actually clump up and group up. Now, AB individuals have both of these antigens on their cell surfaces. So AB patients actually do not make any antibodies. Not against these anyway. Try that again. Antibodies. Because if they made A or B, their own blood would clump, and that would be bad news. Now, O-type patients make no antigens, so they make both A and B antibodies. Because O does not want foreign invaders, right? Don't want anything like that. A and B antibodies. Don't want foreign invaders. Let's see. Check my notes here. So let's take a couple of looks at some images from the text. So here is an image of multiple allelism, and this is figure 920 on page 280. And this talks about how we have 3 different alleles that make 4 different blood types because of their combinations. So we have codominant here with IA and IB, and then of course we have our one recessive allele. So this image here gives us a nice idea of what the multiple allelism actually looks like when we have 2 together in individuals. And of course we can see their genotypes. The phenotypes are the 4 different blood types. So these are the phenotypes. But these down here are their genotypes. Remember, genotype is about the genes. Phenotype is what is physically expressed. Now, we can't necessarily see the fact that they have A and B antibodies or antigens on their cell surfaces, but we can detect them using blood typing kits. So this figure is from your textbook, figure 921. This is on page 281. And this is a really nice representation of what I just put in the table, the A antigens, B antigens, and antibodies. So feel free to look at this more if you like. I really like this figure because it talks about who can donate and who can receive blood. So here's the key to figuring that out. When you think about the donor, so if I'm donating blood, I am type A blood. So if I donate, I have to think about the antigens on the cell surface, right? Because the antigens on my cell surface matter who I can donate to. If you're thinking about the recipient who's going to be receiving the blood, you have to think about the fact that they make particular antibodies or not. Terrible. Let's try this again. Okay, so let's consider this. I am type A blood up here at the top. Now, if I'm donating blood, right, I have A surface antigens. Those are these little things on the cell surface here. Those are my A cell surface antigens. So I can donate to anyone who has those A surface antigens because they do not make A antibodies. So if I'm thinking about the person going to receive that blood, they don't make the antibodies that are going to attack it, right? We don't want that. If I were to donate, say though, to type B blood, they make A antibodies. They don't want that in foreign blood, and so they would clot the blood, and that would be very, very bad for them. Let's not do that. So if I'm thinking about the recipient, so I am type A blood. I'm going to circle up here in green. If I'm going to receive some blood, I want them that either have A antigens or no antigens on the surface so that my body does not attack it. Okay, it's a little confusing, but just remember this. Type O blood is what we call the universal donor because it has no surface antigens, neither A nor B antigens. Okay, so nobody's immune system can attack it. That's great. It's the universal donor. Type AB blood, right above it here, is what we call the universal recipient because it doesn't make A or B antibodies. It doesn't make anything that could attack foreign blood. So they can receive from any type of blood. Pretty convenient. The final thing I want to talk about is about the Rh factor. So this is an additional factor that's separate from the ABO blood type, but determines our compatibilities with other blood types. So let's imagine we have two chromosomes, mom chromosome and dad chromosome. Mom was orange, dad was green. So Rh factor it stands for rhesus factor. If you're interested, it's in the lab. And this was first discovered in rhesus monkeys, and it's just an additional factor that is simple dominance. There is one dominant and one recessive allele. The dominant is positive, and negative, Rh negative, is recessive. So we can have an individual that has, say, a dominant, remember we did this in class. So the stripes means dominant, and polka dots, sometimes it doesn't like polka dots, but it works today, equals recessive. We can have two dominant, and that means the phenotype of this is Rh positive, right? We can have an individual that has is a heterozygote for this. And their phenotype is also going to be Rh positive because they have the dominant there. And we can have another individual who has the both negative alleles and is homozygous recessive. And this phenotype is going to be Rh negative. So this is inherited completely separately. It's on a completely different chromosome, even though the ABO blood type is what we might call it. So this is just an additional surface marker. So for example, I have A positive blood. So I have A antigens on my red blood cell surfaces. So like this. And additionally, I also have the plus markers. If you do not have a plus, if you are, let's say, a minus or a negative, you are not going to have any additional surface markers. So we can continue this. It can get a little bit more complicated, but we have multiple different blood types here. Now, so let's take a look. I'm going to draw 8 blood cells, and this will finish it up here. There, not very evenly spaced, but that's okay. And that one's hidden by the video. I'll try not to do anything there. Okay, so if we have A positive, or let's say A negative, let's do A just minus. A negative blood. They have the A surface antigens, but no Rh antigens. A positive blood has the A surface markers, and it has the positive surface markers. I think you get the idea where this is going. Not very good, but you get the idea. So if they have B type blood, B negative, oh, I didn't plan this very well. Let's do a different color here. B type blood, B negative will have the B surface antigens, but no Rh positive. B positive will have the B surface antigens and also the Rh factor. We can also call it the D antigen. That's another one. AB, of course, we get even more complicated here. AB negative, we have AB positive. So AB negative has both A and B surface antigens. And AB positive has all 3. So, oh no, I did the B in a different color. Trying to be all consistent and then I forget. There's my B surface antigens. Try again. The problem is that I can't go back and re-record sections of this, so I just have to kind of make do unless I want to record the entire slide, which will take a long time. And then finally, we have O negative, which has nothing on its surface, and there's also O positive, which has just the Rh surface antigens. I hope that makes that pretty clear. All right. So one more point about the Rh factor. So the Rh factor individuals, if they are positive, they make no Rh antibodies. So basically anti-Rh. right? We don't want anything to clump to any of these together in the positives. If you're Rh negative, however, you make Rh antibodies that will prevent you from receiving Rh positive blood. Okay, in the next part of the video, what I will be doing is actually demonstrating how to do the blood typing. Thanks for watching.